Hello everyone, my name is Ryan Pianka and I'm the undergraduate lab assistant here at the Ocean Genome Legacy Center. My talk is titled EDTA, Ethanol and Salt, Nuclease Inhibition for Long-Term DNA Preservation, and I'm excited to be sharing it with you today. A little bit of introduction for those of you who don't know, OGL is a genomic biorepository. Our business is in long-term DNA storage. And theoretically, this seems pretty simple, right? DNA is a very thermodynamically stable molecule that doesn't degrade much when it's isolated. However, isolating high-quality DNA is made much more challenging due to enzymes called nucleases. These enzymes are released by cells quickly after death and degrade DNA in tissue before we can even extract it. This is why many field researchers place their tissues in liquid preservatives such as alcohol, as these ideally inhibit enzymes and preserve DNA in tissue for long-term collections like OGL. However, in order to do our work in this age of modern bioinformatics, it's very important that we have high-quality DNA extracts. This is why I posed the research question this semester: which tissue preservatives yield the highest quality DNA extracts through nuclease inhibition? Before I get into my study, first off, we need to define what I mean by high-quality DNA. OGL defines high-quality DNA extracts as containing high molecular weight strands greater than 10 kilobases in length. We see this visualized on this gel right here, where these dark bands are above the 10 kilobase rung of this ladder. Conversely, we see some low molecular weight smearing right here, which appears below the 10 kilobase rung of this ladder. Notice how it appears as a smear because small DNA strands can move very fast through gels. Now, let's talk about the actual enzymes that degrade DNA. In my study, we're focusing specifically on DNase 1, a very ubiquitous enzyme. From cryo-EM studies, we know that this enzyme requires divalent calcium and magnesium for catalysis. This is really important information as we can use it to consider some good inhibitors for it, and thus good preservatives of DNA. So that's exactly what my study did. It looked at 3 different inhibitors / DNA preservatives and compared their effects. So the first one was EDTA, which is a very good divalent chelator, meaning it's a compound that can grab metals. So theoretically, it can grab these enzymatic cofactors and remove them from the active site, therefore disabling the enzyme's functionality. The second preservative we looked at was concentrated ethanol. This is because concentrated ethanol is very good at interrupting the tertiary structure of proteins, or the non-covalent bonds that hold proteins together and enable proper folding, visualized as this hydrogen bond in the green circle. Instead of the properly folded protein on the left, in the presence of concentrated ethanol, we get a protein looking like this on the right. With those circled bonds gone, the protein parts cannot fold properly, and we get a functionless mess that looks like silly string. Lastly, we may be able to improve the other 2 preservatives by saturating them in sodium chloride. This is because sodium ions can competitively inhibit cofactor binding. With the improper ions bound, the enzyme cannot work normally, and thus we lose functionality. So my goal is to compare these preservatives independently and in combination with each other to find the best option for DNA preservation. Here's a schematic of what this looks like. We sampled tissue from 4 invertebrate taxa, and then we were treating this tissue with semi-factorial treatments. For control, we have flash frozen tissue stored at -80 degrees, and then we are testing the following liquid preservatives stored at room temperature: 95% ethanol, EDTA, salt-saturated EDTA, EDTA with ethanol, and salt-saturated EDTA with ethanol. After time points of 6 weeks, 6 months, and 12 months, DNA will be extracted from all these tissues, as well as fresh and freshly flash-frozen tissue for reference. And then we'll analyze the DNA through several methods, but today I will only review gels. Now I'd like to share some of our 6-month data from our blue muscle taxa. In this image, we see what DNA extracts look like from fresh, frozen, and ethanol preserved tissue. We see that there's some high molecular weight DNA, but there's a lot of low molecular weight smearing. Now let's compare this to the other preservative mixtures. We see that storage in EDTA yields high molecular weight bands, but they are relatively faint, indicating a low DNA concentration. However, this banding appears darker when ethanol is added. Darker bands indicate potentially higher high molecular weight DNA concentration. Next, let's review our bristleworm taxa. Like mussels, there is significant smearing across fresh, frozen, and ethanol treatments, but it appears there is more concentrated high molecular weight DNA in the immediate extractions than the ones stored for 6 months. We see something different in our tissue stored for 6 months in EDTA-based treatments. Largely, there is concentrated high molecular weight DNA across all these treatments. However, again, we see that high molecular weight DNA bands were consistently darker in EDTA mixtures with ethanol, indicating good DNA preservation. Finally, let's look at bloodworms. We see a similar pattern to bristleworms, where there is degradation in the flash frozen and ethanol preservatives after 6 months in storage. Bringing in our EDTA-based treatments, we see a reduction in low molecular weight DNA smearing and degradation. Interestingly, EDTA alone produces inconsistent and faint high molecular weight bands, but all the modified EDTA mixtures produce consistently darker bands. We also see that salt may in fact aid in preservation as theorized. The addition of salt produced darker bands in these treatments. These findings are very exciting, and more results will be shared after the remaining taxa and time point data is analyzed. We can't make any firm conclusions on the best preservative combination yet, but we look for the finishing gels, tapes, and spectrophotometry. With this data, we can use statistical analysis to make a conclusion about the best preservatives for high molecular weight DNA. Thank you so much for listening to my talk, and I'd like to thank all of my collaborators and the whole OGL team for assisting me on this project. Their insight and their assistance was invaluable and made this semester an excellent research opportunity. Thank you very much.